So, ayan. Sige. Ayan. So, okay. Okay, so last meeting we were discussing on hemoglobin and how hemoglobin is vital, especially in the oxygenation. And we all know that erythrocyte is transporter of hemoglobin, and hemoglobin is the one in actual saturation with the oxygen. And without the hemoglobin, okay, so exogenations in the body will not be expected. Now, these are the main function. Okay, so as it distribute oxygen or the oxygenations in the whole part of the system, okay, it also in return collect and waste products. And one of the waste products collected by hemoglobin is your carbon dioxide. And with this carbon dioxide, it also degraded, okay, to produce your hydrogen ion and bicarbonate, which are very vital, also not important in the blood buffer system. So for the synthesis hemoglobin, now I want you to remember how important each component in the synthesis of hemoglobin. As what you want to remember the erythrocyte, kasi di ba there are part in the erythrocytes which are very crucial, especially in the sequential. Yes. Okay. Yan. Okay. So the sequential synthesis of hemoglobin also is very important. Now, if you will notice glycine plus succinylcholine-CoA, are you familiar with these two substance? Where did you first encounter the glycine and succinylcholine-CoA? Saan nyo na-encounter yan? Anyone from the group? But this is a part of the Krebs cycle. Now, if you still remember the cycle in your citric acid cycle, you will notice glycine and succinylcholine as one of their byproduct and substance or element that are very important to complete the Krebs cycle. Now, in that portion, no, so a portion of that are very important in the synthesis of hemoglobin. And in the presence of your 5-ALA synthase and your B6, so I hope you still know what vitamins is B6, no? So without this, there will be no generations, okay, or synthesis of your ALA. Yan. So this ALA, yan, so two molecules of ALA with the activity of an enzyme for the 4-phosphorylinogen synthase. So this is very important, no? Will produce the 4-phosphorylinogen. Now, if you still remember during our general pathology, there is this conditions called porphyria. So, can you still remember how porphyria or the characteristic of porphyria in affected animals, or how these pigments, actually porphyria is a pigmentation and it has a reaction to UV light. Okay, so can you still remember how this porphyria affect the animal? So what do you remember in our discussions in general pathology with regards to porphyria or pigmentations like this? Hello, naririnig niyo ba ako? Ito ayoko pag nag-discuss online. Can you open your cam, all of you? So that I know that you're listening to me. Kasi alam ko may mga activity kayo this afternoon eh. I don't know if you're still listening to me or not. Okay, so what do you remember in porphyria? Okay, so what do you remember about porphyria? Why am I asking about porphyria? Now, I want you to connect now how porphyria become an element in the conditions in the pigmentation. Okay, ito yung porphyria is the condition. The element involved or the material involved or substance involved in porphyria is the porphyrin pigments. Okay. Now, porphyrin is an active form which is being synthesized from the two ALA molecules. Now, with this porphyrin, okay, so this will give off 5 porphyrin reactions. Now, remember this porphyrin reaction. So, that means that a particular part, once hemoglobin is degraded, ito yung application niya or this will be connected in the porphyria conditions. Kasi porphyria, in some way, nagiging pathologic siya because of its, what do you call this one? Interactions with the uvuli. So, ang kanyang manifestations in the skin once porphyria is positive, no? Or let's say, seen, okay? Sa condition, nagkakaroon siya ng parang parang masusunog. That's how it will react to UV light. Yeah. So, nagkakaroon tayo ng hypersensitive in the skin or yung hyperkeratosis also. Ayan. So, please do it on that. Okay. And then, of course, with the porphyrin reactions, that will give off photoporphyrin. Now, this photoporphyrin, 9 plus porphyrus. So, this is how iron is included in the synthesis of hemoglobin. That's why, in the absence of iron or deficient iron, it will not only affect the erythrocyte erythropoiesis, but also with the hemoglobin synthesis. Now, with the iron or the ferrous, okay, the ferrous state is the active state where it can be used up, okay, in an activity or na pagtapolate activity. Yung ferritin kasi, yan. So, these are the stored form of hemosiderin are stored form of iron. Okay, so with the presence of sulfuric helicase, so this is the rate-limiting enzymes that will facilitate the formation of heme. Now, heme now is a part of the hemoglobin, but hemoglobin is not complete because heme is only produced. So we need also the protein. Okay, now with the heme, this will be transported or this will be synthesized also to become ferriheme. So these 4 ferrihemes has 4 molecules of iron plus 4 molecules of your your protein, your globins, no? That will make up your methemoglobin. And this methemoglobin in later part become your hemoglobin. So chances that let's say conditions affecting the erythrocyte production as well as the degradations of the erythrocytes will affect the activity of your hemoglobin synthesis. Now, when this hemoglobin synthesis takes place, so once erythrocyte is being produced, okay, so hemoglobin also follows the production. So they are simultaneously produced or developed during the erythrocyte development. Yeah. So please do be familiarized with the important elements that are included, okay, or that are vital in each steps in the synthesis of hemoglobin. Okay, and these elements also are very important, okay, especially in the degradation of hemoglobin, in which once this is liberated, you will notice that a portion of its byproduct becomes bilirubin, right? So, where this bilirubin came from? So, those are some of the questions, okay, that can be answered through understanding the synthesis of your hemoglobin. Now, for the liberations of your, or in the destructions of your hemoglobin, you will notice that the heme is the portion, okay, the heme and the globin. So, alam na natin that in the hemoglobin degradation happened also simultaneously with the erythrocyte destructions. So, that will give off 2 important components, the heme and the globin. And the heme will further be synthesized, okay, to become a biliverdin. Of course, this will also produce a byproduct of iron or the ferrous, and of course a carbon monoxide. So this biliverdin now, through the actions of mga enzymes and conjugation, okay, in the liver, okay, this will be distributed in other portion of the body, especially in the colorations. Ayan, if you still remember your urochrome, so a portion of the bilirubin are incorporated in that. Okay, in the stercobilin, so that is a specific bilirubin pigment that color, okay, or the give of color to feces. Okay. So this virus now is recirculated. Remember that your iron molecules or your iron are not excreted. Okay, they are stored or reused. Now, syempre, your carbon monoxide is a waste product. Now, for the globins, so since globins and amino acid, they are recirculated. And all know that globin are part of the plasma proteins. And so, you need to remember that. Okay. So as for the iron fate, so I think I have mentioned about the fate of iron last time, but for your remembering, no? So we have three major sites where iron are being stored. So I just told you about iron which are not being excreted. So they are not excreted. Okay, they could either be reused or stored. So in that matter, no, so we have three fate for the iron. So about 50-70% are part of the hemoglobin, okay, the erythrocyte hemoglobin. And the 25 to 40% are stored as ferritin or hemosiderin. So this ferritin and hemosiderin, once needed, no, there will be recirculated and being used up. Okay, and then the remainder of that, so ilang percent na lang natitira dyan? 5, 60, 70, 40. Parang masyadong sobra tabsa. Okay, the remaining 5 or lesser, so those molecules are used. Okay, the pigment, okay, of that, the pigment liberated from the iron will be incorporated in the muscle. Okay, so as to become a myoglobin and other color of parang color pigments. Yeah, so that's why part of the cytochromes and part of an enzymes. Okay, so these are the fate of iron. So you can read more of this from Stockholm. So if you have the Stockholm, okay, si author niya sa Stockholm, no, for the clinical, okay, pathology. So just do the important what you need to remember, okay, is how iron regulated, okay, and used up, okay, and how vital iron in the development and productions of your erythrocyte and hemoglobin. Yeah. So also please do read on the ceruloplasmin. Yeah, this is one important copper containing copper containing protein, okay, which are synthesized in the liver. They are very important, okay, as a necessary transporter to iron. Okay, so now we go to the erythrocyte evaluation. So particularly on the erythron. So I mentioned about erythron during our first meeting as the mass, okay, of the circulating erythrocyte together with the substances, okay, included in the composition of your erythrocytes. So how important, okay, how important the diagnosis of CBC or the diagnosis of blood, okay, for the disease diagnosis. Ayan. So, the evaluation of blood smear and complete hemogram is used to better understand disease mechanisms, particularly in hemopoietic system. Sorry, dito pala yung. Okay, particularly in hemopoietic system. And when we say hemopoietic system, this majority includes the blood. Okay, so the blood dynamics as well as those blood, okay, forming organs. So these are important. So in our systemic pathology, we mentioned about other organs, okay, that completes hemopoiesis. So these are important. However, in our discussions clinical pathology, only the bone marrow is included in here. And the other the other organs, they are somewhere not directly, okay, mentioned in this part, since the laboratory or the laboratory part or activity focus on the blood. Okay? So pathology of the RBC mainly in the area of production, morphology, and the number of cells and the nature of hemoglobin. That's why in the laboratory activity, we will discuss or we will focus on how how blood is assessed, okay, quantitatively and qualitatively. So in what you see in the picture, okay, the picture shows you here the blood smear and capillary, okay, as well as the blood collected. So all of this, I hope you will do is you can do it, no, in your laboratory. Okay, so just do remember that when you collect blood, avoid hemolysis because hemolysis will mask, okay, the really picture, okay, of the blood component as well as the blood values that you will be expecting to read. Okay. Another is avoid using syringe that contains water and chemicals that might destroy the RBC. Okay, so ibig sabihin, you can go back to the concept of the hemolysis. So always remember that avoid hemolysis of the RBC. So always use fresh blood if possible, but in times that you need to store, okay, a portion of blood, so you need to consider how long it will be stored. Ayan, kasi most of the time it should be not more than 24 hours na naka-refrigerate. Okay, so hindi to frozen ha. It will be under refrigeration but not frozen. However, there are chances that the morphology, okay, or the integrity of the RBC uh, um, collected, okay, the blood collected can parang ma-reduce. Ayan, kasi hindi na siya fresh. Now for the tube that you will going to use, at least use heparinized tube. But that depends on the focus of your examination. Okay. Then of course avoid contamination. So always remember that in doing everything not only for the blood collection, okay, you avoid contamination. So if you think the risk of contamination is high, then do all the necessary, okay, the necessary things that you could lessen the possible contamination, especially if you need to get samples for microbiology. So yun yung mga tatandaan nyo. If meron tayong contaminations, then you should consider that during your interpretation or sample interpretation. Okay, so I want you to read on the smear preparation. So this is a part of the, I think it's for the laboratory. So I want you to read on the smear preparations, particularly the wet, thin, and the thick smear. So of course, please do read also yung mga required volume of blood to be collected for hematology. So you can get the references in SCALM, okay, blood, okay, yung sa hematology. Yan, you can read on that. And then, of course, site of blood collections of any punctures common animals, particularly those domestic animals that are assigned to you, and common anticoagulant use, and of course, some basic information in regards to the lifespan of RBC sa common domestic animals. So please do read on that. Okay, now we go to the erythrogram. Now, the erythrogram composed of three important elements. So we have here the RBC count, the packed cell volume, the hematocrit, and of course, the hemoglobin concentrations. Now, if you will remember yung nabanggit ko kanina, so I just mentioned about that, no? Those possible assessment that you can expect for the blood evaluation is the morphology, and the morphology, the number, and of course, the hemoglobin concentration. And those are actually reflected in the erythrogram assessment. So that is your RBC count, hematocrit, and hemoglobin concentration. Okay. At the moment, no, there is an automated way of evaluating now the erythrogram. So since we have the hematology analyzer, in which you can just put a sample of blood in a tube or special tube where this equipment was provided, no, then it will be read. And then the result will come just after, siguro a second or after a minute, no? Quantitatively. Yan. The thing is, the qualitative component should also be evaluated. That's why the manual evaluation or assessment, you do the blood smearing. Okay, so you can see how, okay, the, let's say the morphology, okay, of blood will look like after. And actually, this can also be related in the quantitative result, okay, that you can get from the analyzer. Okay, so let's start with the total erythrocyte count. Now, the aim for the total erythrocyte count is to enumerate, Okay, the total count of RBC per cubic millimeter. This cubic millimeter is how you're going to read on that under the microscope using the Neubauer slide. Okay, so the indications to conduct total erythrocyte count is to determine the functional state of erythron. So since we talk about erythrocytes and its component, again, so the erythron there is included, especially in determining once you do interpret the result that you can obtain. Now, determine the presence of intracellular parasites. Now, for the presence of intracellular parasites, so especially if you can do this with simultaneous with the blood smear. Ayan. So you can see whether intracellular parasite is present, or for the quantitative effect, okay, the quantitative result, sorry, for the quantitative result, so you will correlate, okay, the value, okay, the value that you can obtain from the from reading of the total erythrocyte count. So aside from that, blood glucose levels also be incorporated or as related, okay, in the total number of erythrocyte count, and particularly the state of nutrition. So in the total erythrocyte count, you can list also the possible condition of the animal, whether the animal is malnourished, or at the same time, you will notice if it is anemic or not. Okay. So for the methods, okay, for the total erythrocyte count, okay, we have three. We have the hemocytometer method, the electronic counting method, and the photoelectric method. Thank you. Okay. Yeah, so we have the hemocytometer method, the electronic counting method, and the photoelectric counting method. Now, the two there, okay, the last two there is more of parang automated. Okay, as you can see, electronic and the photoelectric counting method. Now, we have this hemocytometer method in which you will use, okay, a Neubauer slide, a special pipette, ayan, and then of course, yung microscope where you can count individually, okay, the distribution of RBC in a sample. Okay. So we can do that. So I hope you can do that here. So ito yung isa sa mga activity natin in the laboratory. Ayan. So for the hemocytometer method, these are the basic equipment. So we have the light microscope or light microscopy. Okay. So I think we have the binocular naman. Okay. And then of course the hemocytometer. The hemocytometer will include the Neubauer slide. Ayan. So you will notice we have the Neubauer slide here. Ayan. Parang it resembles also. This is also the same as the slide that you're going to utilize in the egg per gram. Okay, dun sa parasite, okay, sa parasitism, sa fecalysis. Okay. So other than that, we have the diluting pipette. Ayan, so special pipette for the RBC. So only the red one, okay, yung may red bead. So those pipette with red beads, yan, these are the important pipette for the RBC counting through using the hemocytometer method or hemocytometer method. So we have the cover slip, special cover slip, medyo makapal ng konti, and of course, the reagent. So the reagent used here, so we have the isotonic, yeah, the isotonic of fluid, ayan. And of course, other name is your Hayam solution. So yun yung mga use na diluting fluid, okay, for RBC. So remember, di ba, ang RBC is very vulnerable in the changes of, or to the changes of fluid, okay, its environment. Baka magkaroon kasi ng changes in the morphology, at the same time, it might hemolyze, okay, the sample. Okay, for other information, so these are the red bean for RBC and the pipette with a with a dilution of 1:200, where blood is drawn to 0.5 mark. So this one, so you will notice you have the red pipette here. This is the calibrated. So this is actually a graduated pipette. So to this point, you can draw or aspirate the blood using a pump, a specialized pump to, or yung iba, pwedeng sipsipin. Okay, what we did last time is pwede nyo siyang sipsipin. Pero syempre, ingatan nyo lang, it should be until 5. It should be until 0.5 unit only. Okay, and then afterwards, no, you aspirate the dilution. Okay, yung diluent mo. Okay, so yung agent mo until this 1:1 mark. So, dito, afterwards, no, you need to shake. You can buy now, there's a shaker. Okay, there's a shaker now for RBC pipette. Okay, so in UPLB last time, when I'm doing my MS, no, I study, no, so hindi na siya manual, just like 8, just like that, no. So, ilalagay na nila dun sa shaker for a particular minute, mga 1 to 2 minutes, ganyan. Afterwards, yun na, nakaano na siya, na dilute na siya. So, dun ka na magbibilang. But they do also the manual cutting, okay, under the microscope. So, afterwards, no, pag na-delete mo siya and shake mo na siya, you can now put that here. Okay? Meron naman siyang indications dito. Ayan, dito yung mga counting grid natin. Okay, so you can dispense the fluid, okay, on this part. Ito yung canal. Ayan. Ayan. So, and then you can put on top the special cover slip. So, this is how it looks like. Okay, in here, itong dalawang part na to. Okay, this is how it looks like. Okay, so you will now start to read on the diluted or distributed RBC, okay, on this grid. Okay, itong 5 grid na yan. Yan. So, cell touch in the top ruling in G should not be included. Only those inside. Yan. So, the way you're going to read on that is ito, for a very systematic, for a better systematic reading. Yan. So, under HPO, counting in the 5 smallest squares on the center by the counting chamber, then you will multiply at 10,000 for the dilutions. Okay, so ganyan yung kanyang lalabasan. Then, on this part kasi, yung other portion of the square, yung outside of the square, okay, this is where you're going to read on for the leukocyte count. So, isa din lang. Ito rin ang gagamitin mo. Okay? You need also to use the Neubauer slide for the hemocytometer method. Okay, so that is your hemocytometer method. The thing is, the error might be originated from the one counting. Okay, so medyo mataas ang error dito, lalo na kung hindi mo makita yung RBC. Okay, and then nagkamali ka ng counting, doon nagkakaroon ng error. So this is one of the disadvantage of the manual counting. That's why most of the activity now is done through electronic counting method using hematology analyzer. Okay? So this could give more accurate counts that can be attained by hemocytometer method, and that this should be optimized first. Should be optimized first. So hindi nyo gagamitin yan ng walang optimizations. The optimization is that you should optimize the number standard values that you can use, no? And of course, how this value can be evaluated. Kasi meron naman yan, is automated. You can just put on it, may software siya, papasok mo lang siya, and afterwards, kapag nag-run ka 1 or 2 for optimization, pag pareho sila at maganda yung lumabas, that's how you're going to optimize, and then you can now read your sample. So, walang variation. So, this is more accurate, okay, than the manual counting. And then, ang ibibigay na lang sa'yo is the result. Okay, it's already printed. Okay, printed result. Yeah. Okay, for the photoelectric counting method, what you read on this is the light. Okay, the light emitted. Okay, the light emitted, which is transmitted through a suspension of erythrocytes in the diluents. So you can just put sample from a certain slide and then put that in a particular counting method, and then yung light na to ang magre-read. Okay, so para siyang a concept of this spectrophotometer. Okay, so ganun yung kanyang concept. Okay, now, so those are part of the total erythrocyte count. Okay, for the total RBC count, so we have the hemocytometer method, the electronic counting method, and the photoelectronic counting method. Okay, now for the hematocrit. So this is, this can be done here in our laboratory since we have the centrifuge. Okay, so what you check for the hematocrit is the percent of blood composed of erythrocytes that is readily accomplished by the centrifugation. So what happens here is that the most dense, okay, which are the erythrocytes, are sedimented, or they are at the bottom. They are packed in the bottom. So these are the one, or these packed erythrocytes are called the PCB, is the one that you will going to read using a reader or a graduated reader, okay, in percent. Okay, so I don't know if this is your first time to see a hematocrit tube. Okay, so the parts of the hematocrit is the PCB. This is where our focus on the puffy coat and the plasma, or the blood plasma. So the puffy coat is white or gray layers of thrombocytes, and of course other WBCs can be seen also in the puffy coat. However, there are also some parasites, okay, where they can be achieved, okay, or seen in the puffy coat, like the trypanosoma. Okay, and of course the blood plasma. So the plasma is the fluid portion of the blood. Sometimes the color of the plasma varies, no, depending on the hydration and the status of blood, okay, in the of the animal. And so this is one example of hematocrit reading. So you have the normal blood. So this is in human, no? So in human to. So we have the blood component, okay, particularly in the hematocrit component, or reading for a female blood, that is 37 to 47% of hematocrit. So if you get that, so nagpo-fall under or nagpo-fall between here, then you are in normal value. Okay, so nasa normal range ka. So same with the male, okay, 40 to 52% hematocrit. Now for the anemia, syempre lower than this one, expected to be that portion. And then polycythemia is higher than the expected normal. So the key for the interpretation, especially of this kind of interpretation, is the reference value. So for the reference value, you need to consider where you get the reference value. So I'm always mentioning this one to not only to your class, no, but also with the other one. Kasi kailangan yung i-relate that where you get the sample, the same time yung environment. It might be different. Okay? So sa parang tropical countries and the temperate countries might have a simple variations. Pero kung papasok siya in the range, wala namang problema. Okay, but if you get reference value, much preferably ay yung malapit dun sa conditions where you get the sample. So kung meron ng standard value from the parang conditions here in the Philippines, then much better. Now, kung wala naman, we still opt to use other values, no, or the standard values, which could be applicable both in temperate and tropical, then pwede yun ang kunin natin. Okay, as our value during our intervention. So it's very important. Okay, now, for the types of hematocrit determinations, we have here the macrohematocrit and the microhematocrit. For the macrohematocrit, we use here Winthrop method. So the Winthrop method here is using a specialized graduated tube. So you have that tube, no? A graduated tube. Okay, na may reading din siya. Okay, so may reader din siya. So you have here the examples, okay, or how to perform the macrohematocrit or the Winthrop method. So you get or withdraw the blood, okay, and place the specific tube, and then you centrifuge the sample. So always remember that if you do centrifuge, should be in duplicate. Ayan, para dun sa balance for centrifugation ay maganda. Kasi kung hindi, puputok yan. Okay, of course, in the layers of Winthrop tube after centrifugation, the same, you can get the plasma, the buffy coat, and of course, the hematocrit. Then we have the readers for the Winthrop's method. Okay, so meron tayong mga graduated number or measurement. Okay, where you can check this one in percentage unit. That's why I want you to know the unit, the unit measure for the assessment ng mga values. Okay, kasi napaka-importante na. Okay, for the microhematocrit, oh, by the way, we have this kind of centrifuge here. I don't know if you have already seen this one, but meron tayo nandito na, binaba na dito. So, meron yan. Ayan. So, the other one is this capillary. That's why it's called the microhematocrit. So, this microhematocrit uses a capillary tube or capillary hematocrit tube, which approximately 7 centimeter in, or 7 centimeter long. So this is the common, okay, activity done, okay, in hematocrit determination by using your capillary tube. So sabi ko sa inyo, dapat meron kayong capillary, nakabili kayo kasi yan yung gagamitin nyo. Not only for the erythrocyte assessment but also in clotting assessment, sa clot assessment natin, sa ACT natin. Okay, so same as this one, we have the specialized reader. So we have this kind of centrifuge with a specialized reader. Ayan, makikita nyo siya. So, how will you going to check for the measurement? So, dun sa capillary nyo, you need also to have the Adams clay where you can put that below dito para hindi lumabas yung blood. Okay? So, yun yung magiging reference nyo. This is the point of reference pataas. Okay? And then, look for the corresponding number in percentage. Okay? So, hanggang doon yung paxel. Do not get all the component, only the paxel layer of the RBC. Okay, now let's see the advantages and disadvantages of conducting hematocrit using a microhematocrit technique. So, advantages, it requires small amount of blood. So, ilang ml lang yan. Wala pang 1 ml. Okay, less than. So, you can also conduct that in less time kasi mas maliit. Okay, diameter. And if you're going to centrifuge that in a matter of minutes lang, okay, matagal ng 5 minutes, no? You can get the pack, okay, of the RBCs and the other component. And then the results are more accurate and reproducible. So since small lang siya, and then malilimit mo yung chances of error. Okay, and then of course you can reproduce that, yung duplication, okay, to see the validated result afterwards. Now, the disadvantages, you need a specialized reader that could read on that kind of hematocrit. Okay, ESR can be determined because of the maliit masyado yung capillary. But anyway, in the ESR, you cannot, you are not going to use that kasi mahirap talaga. And the difficult to evaluate the depth of the buffy coat, which sometimes you cannot see the buffy coat on that particular map kasi masyado siyang maliit. Okay, yung gray in between. Minsan na lang ang nakikita is the paxel and the plasma. And the part of the buffy coat, hindi na masyado makikita. Lalo na if there is leukopenic, okay, leukopenia. Yan, so hindi na siya masyadong makita. Or minsan wala na. So, that is one consideration pag wala nakitang WBC, no? So, yun yung pwede mong i-list as possible. Okay? This, no? Especially if it is related to the clinical manifestations that the animal is present. Okay, now hemoglobin determination. So that's how hematocrit is being conducted and how it is being interpreted. Okay, for the hemoglobin determinations, you have these reflections of the ability of the erythron, particularly the erythrocytes, okay, to carry the oxygen. And this provides the most direct indication of oxygen transport capacity of the blood. Siyempre, we are determining the saturation. Okay, so in general, so mammals, mammals, in general, it should be 1/3 of the PCB of erythrocytes are of normal size. Okay, so normal size, you can check on this on the smear, pag nag-ano ka smear. But definitely, you can check this during the total count, okay, kung normal siya. And there are some related erythrocyte indices that could determine, the normality dun sa size. Kasi later, we will talk about the MCH, MCB, and MCHC. So, which is also related in the erythrogram result. So, it helps you to understand more on the erythrogram result. Kaya, if you are familiar with your CBC result, kung meron kayong CBC result, you now check for those different element or erythrocyte indices and erythrogram parameters. Ganun nakalagay doon. So, it should be, para mas maintindihan nyo siya, you can parallel. Okay? You can parallel and then check whether pareho sila or may konting variations. And that can give you simple differentials, okay, on that. Okay, so for the methods for hemoglobin determination, so we have here the acid hematine method, direct matching method, oxyhemoglobin, the cyanamide hemoglobin, and approximation method. Now, here, so since CBC, hemoglobin, and other components of the blood can be run in one activity. Ayan, using the blood analyzer, madali na lang siyang makuha. But before, no, we did also the manual counting or the manual, assessment using the SALI method. So this is the SALI method, we have the comparator plot. We still have this one, but it's not functioning anymore. Aside from that, wala tayong reagent. Okay, yung specific reagent na timpla. Okay, for this one. Kasi ito nabibili na lang. Kung magkamali ka kasi sa pagtitimpla, wala nang matitira. Puro hemolyze na yan. Or let's say, ma-hemolyze naman talaga, but definitely it might affect the interpretation, okay, of the parang colorimetric interpretations with read in a graduated tube, and then para mo siyang compare on the colorimetric plot. Yeah. So that's how the acid hematine SALI method is conducted. So this is depends on the conversion of your hemoglobin to acid hematic, which is usually accomplished by the use of hydrochloric acid. So remember, your HCl is quite corrosive and medyo masyadong concentrated. Yan. So, ayan yung kanyang reagent. To be able to hemolyze the RBC, and the hemoglobin will be liberated. Okay, so that is how it's happened, the acid hematine method. Ito yan. Ito yung comparator block. Dito yung sample. Ito yung reference value mo. So, may mga kulay-kulay yan at magpapalit. So, may reading yan. Okay? So, for direct matching method naman, we have the calculus hemoglobin scale. Colorimetric din siya. And this colorimetric, meron din siyang particular corresponding number. Okay, this is how you're going to interpret, okay, for this direct matching method. So, a drop of blood is placed on a filter paper and then allow it to dry and compare to the color standard. And that color standard will give you an interpretation. Okay, so there is this what we call mga colored scale. Ayan. So, ito yung method na to for screening purposes. Kasi, as you can see, no, you use colorimetric method. So, it's not a diagnostic technique, but more of the screening technique. So, if you want to more accurate on this one, you have the hemolyzer, okay, the hematology hemolyzer, which is more accurate, okay, in determining the method, okay, for the assessment for the evaluation of concentration, hemoglobin concentration. Okay, another is the direct matching method. So, using a direct hemoglobinometer. Ayan. So, ito yung sa hemoglobinometer. However, it's quite obsolete according to references. So, hindi na masyadong ginagamit ngayon. Ayan. So, this is how it looks like. So, meron tayong visualization. Kung naalala nyo kanina, a drop of blood in the photoelectric method dun sa erythrocytes. So, it resembles like that, yung kanyang concept, because you will notice the illumination, okay, from that sample of blood. Okay. So, yun yung ilalagay mo sa specific area. Okay, dito mo siya ilalagay. Then, titignan mo yan. Okay, titignan mo siya through this one. So, that will give you a scale, okay, for that particular reading. Ayan. So, may small glass chamber. Okay. This is the pipette here and the apertures. Ayan. And then, the best one, ito yung isa sa mga ginagawa din. Okay. The oxyhemoglobin method using a Spencer hemoglobinometer. So, dito, color standard din siya. However, we have the graduated. Okay, meron tayong graduated comparison. Ayan. So, you put a portion of blood in here and then red, okay, using the intensity of green color. Ayan. So, ayan, may green filter din siya. Yun yung titignan. Okay, this is how it looks like. Then, the absorbance. Okay, the absorbance of the oxyhemoglobin, the oxygen and the hemoglobin saturation. Okay, now this is among them is the best method. It is a cyanamide hemoglobin. This is being used as an accurate method in the determination of hemoglobin concentration, the cyanamide hemoglobin. As you can see, there's a use of the spectrophotometer. So very specific to, it's a quantitative. You just put the specified sample with the cyanamide hemoglobin. Ayan, the drop skin solution, okay, which contains your potassium cyanide, okay, cyanamide hemoglobin. And then, of course, the potassium ferricyanide are the component, okay, that will determine, okay, this solution will determine the hemoglobin oxidation. Okay? Ayan. So, dito mo i-read. This is more of quantitative. Ayan. Kaya mas accurate siya. Okay, so I think we end this our discussion this one. So please do read on this one, basic compass hemogram, including the indicators of the hemogram measurement, component measured, and units. I think some of you included this one in the binigyan yung activity. If not, no, please do include this one as part of your home assignment. Okay, and then look for the other methods used in estimating hemoglobin, especially on the advantages and disadvantages. So remember, we have 4 methods. Now, if you can get other, kasi in human meron siya, and then please meron yan, may nakita na ako sa research, you look for the other methods in the estimations of hemoglobin. And then all answers will be in the tabulated form. So next meeting, we will discuss on the erythrocyte indices and the rest. Okay? So can you still hear me?
Yes, pwede.
Okay. So yun na yun na. I will, sige na, i-upload ko to sa inyo para meron na kayong babasahin, ha? Okay, and then of course, para meron na kayong reference. Okay, so see you next meeting. So I hope you learn something from our discussion. So napakakunti lang yun. Okay, hanggat wala si Doc Mifo, ako pa rin muna maghahawak sa inyo, ha? Okay, sige. So if you don't have any questions, you can reserve that and question me on Thursday. Okay, so see you guys and good morning. Don't forget your attendance, ha? Don't forget your attendance.